two answers to that. That are definitive that scientists deliberately created them. Unless he was a super genius and 10 years out of his time. It definitively existed long before molecular cloning. That's point one. Point two, we know the full sequence. The genome of HIV was published by our lab in 1985, from Paris, around the same time. The genome has no homology to any known existing virus in the world, except SIV discovered after it. Has nothing to do with cats, has nothing to do with chicken sarcoma viruses. SV40 is a DNA virus that comes from little animals that can transform cells in culture, has no sequences in HIV. Further, we've never worked with SV40 with those viruses together, and if we did, the whole thing would be irrelevant. And I think you need to begin with Biology 101 high school. Okay?
These documents, including the U.S. government contracts, best explain how AIDS emerged on two far-removed continents in black Africans and gay New Yorkers by 1978. These were precisely the populations that received the first hepatitis B vaccines, produced in Litton's chimpanzees just 4 years earlier. The man sitting to Dr. Robert Gallo's right is Dr. Jonathan Mann. At the time, Dr. Mann was the AIDS czar for the World Health Organization. Not long after Dr. Mann witnessed my exchange with Gallo, he quit his most esteemed position, saying, quote, far more than a medical problem, AIDS is a socio-political imposition, end quote. If I am right, and these documents are legitimate, then generally trusted vaccines, the sacred cow of public health, given mostly to our nation's youth, said to have contributed most to conquering infectious diseases, are alternatively the world's worst curse and nightmare. The final document in this stunning from the same National Cancer Institute and National Public Health Service report. American biologists and virologists transferring the most sophisticated bioweapons of mass destruction to Soviet scientists during the Cold War. Even though, as Dr. Kano accurately states, the Russians were developing their biological weapons programs at rocket speed. These documents provide conclusive evidence that the NCI's molecular biologists, including Dr. Robert Gallo, America's most advanced secrets in bioweapons production with precise methods and materials for cancer induction, genocidal weapon preparation, and bacteriological bioterrorism with their Russian counterparts and others during the Cold War, for the Gulf War, called Gulf War Syndrome. The Senate thoroughly investigated this subject in 1994 and published proof that our troops were unwittingly used experimentally or alternatively targeted for disease and death by the vaccine makers with official political consent. Spreading across America and the world now is a flu-like illness that resists standard medical treatments. You probably know people suffering with severe recurring respiratory ailments. Experts have linked to an agent called mycoplasma. In fact, chronic fatigue, low body temperature, and a flu that keeps coming back is associated with this germ isolated and patented by the United States Armed Forces Institute of Pathology in a patent shown here. This agent, mycoplasma, apparently contaminated the anthrax vaccine given to more than 500,000 Americans that served in the first Gulf War. Dr. Kano from the Centers for Disease Control applauds this invention. This next government document was obtained by lawyers for the victims I just described. You can see the array of biological weapons being tested on the prisoners in Texas as overseen by Baylor College of Medicine. Baylor, at that time, boasted having George H.W. Bush serving on their board of directors. The dates listed here also coincide with the alleged transition of America's offensive biowarfare program to defensive. Notice the mycoplasma. Both defensive and offensive studies were conducted. A vaccine study and a Hall study, in which prisoners were actually exposed to this airborne agent